Joining us next is also a part of the Danaher group that we're now a part of as well. John Harmer, who is the head of sales and marketing at Vanrx, which is now a part of the Cytiva family. He has over 30 years of experience in the pharmaceutical industry, involved in just about every aspect of drug development and manufacturing. So John's going to talk to us about the aseptic filling solutions that he and the team at Vanrx have developed. So thanks, John.
Thank you, Josh, for the introduction. I'm delighted to take part in the symposium, and also, of course, thanks to my colleague Joe for the very interesting presentation that he just gave. To introduce myself, I'm John Harmer. I'm the head of sales and marketing at Vanrx, which of course is now part of Cytiva, has been mentioned. And also, obviously, with PNI, we're part of the larger Danaher Life Sciences group as well. I'm going to start by providing a brief overview of Vanrx, our design philosophy, and our products. And then before I proceed with some specific considerations for filling mRNA or other lipid nanoparticle drug products. Cytiva and Danaher Life Sciences are unique for being able to support companies from their ideas during drug development through drug substance manufacture and into the delivery of the drug product via injection. So truly an idea to injection process. We were added in February of this year to add to the drug production capacity within Cytiva and Danaher. Our aseptic filling work cells can be provided as part of Cytiva's Flex Factory or Cubio, which you heard about in the previous presentation. And they complement the production of antibodies, viral vectors, or mRNA, as you saw before. Oops, sorry. Got to stop moving my hands so much. And of course, the work cells are available separately for companies looking to add aseptic filling capacity to their existing facilities. Vanrx aseptic filling work cells have been installed all over the world by a wide variety of pharmaceutical companies and CDMOs. Leading mRNA companies such as BioNTech, Moderna, and Genentech Roche have selected Vanrx aseptic filling work cells for their clinical and personalized medicine programs. Most of the installed work cells are being used for developmental and clinical programs, but there are a growing number of commercial products being filled on Vanrx work cells. And there's more on the way. We have 2 customers that have recently completed pre-approval inspections with the FDA that resulted in no observations, in other words, no 483s. One question you may have is why they're called work cells and not systems or filling lines. The reason is that they have a fundamentally different design from conventional filling systems. When the company was founded in 2008, our founders realized that the pharmaceutical industry wasn't well served by the current technology. So they took inspiration from lean manufacturing principles and specifically the semiconductor industry. In semicon, facilities are filled with banks of identical, independent work cells that can operate in parallel. Their capacity can be split between different product lines, and if one work cell requires maintenance, it doesn't stop the whole operation. It's much more efficient than a single large production line. You're able to mix and match capacity to meet your requirements. So our idea was for an aseptic filling work cell was to learn from that example, to get rid of everything that was extraneous and focus on critical requirements. We didn't want to be tied to old ways of thinking, and so we rebuilt the aseptic filling process from the ground up. We intensified the process and used innovative technology to drag pharmaceutical filling into the modern age. And that's a pretty bold statement. Let's have a closer look at how we did it. So this is the interior of our SA25 aseptic filling work cell. It probably doesn't look like any other filling system you may have seen before. The most notable aspect is the lack of glove ports, which means human operators have been removed from the process. In conventional systems, the role of human operators is to monitor the process, waiting for something to go wrong, and then try to intervene in the process to try and correct it. That's not very efficient. In addition, humans are by far the largest source of potential contamination. And the farther you can keep them away from the process, the better it'll be. So we've replaced human operators with robots. Removing the operators meant we had to design a more reliable process, designing the most common risk points out of it. So 2 technologies that support this goal are single-use consumables, like the flow path you see in the picture, and pre-sterilized, ready-to-use containers. And you'll see them in nests and tubs there. And so this is an example of process intensification. All product contact parts are sterilized somewhere else by the suppliers, removing the need to perform these activities on site and allowing the work cell to focus on the critical functions. The use of ready-to-use containers enabled us to make a standard work cell. Because they're supplied in standard tubs and nests, we can use basically the same tooling for any type of container. And keeping them in nests greatly reduces or eliminates the risk of container breakage or jams or process upsets. And so using this approach, the same work cell can fill a wide variety of container sizes and formats without requiring any customization. So let's have a quick look at the products before we get into some of the considerations for LNP filling. We'll start with the system in the middle. The SA25 is designed for companies that need to fill a variety of products in more than one format. It can fill vials, syringes, and auto-injector cartridges with very little changeover between formats. Very little time is required to change over. And it's designed to be future-proof. If one drug candidate isn't successful, it'll work for the next one as well, because it's so flexible. And again, flexibility is built into the system. Our goal was to make, a standard system and to meet 90% of our potential customers' needs with one system so that very little customization is required. And then again, the liquid lyo system is an extension that can be added to an SA25, enabling the production of freeze-dried products. And then if you look over there, the microcell is designed for the rapid turnover of small batches. So it's the perfect system for preclinical, early clinical, but especially it's been designed for personalized medicines. It has an extremely short batch cycle time, allowing the production of many individual batches in a day. So it's optimized for rapid turnover to produce many, many batches in a day. If you have to produce for individual patients or small batches for specific patient populations. So it ties in very well for mRNA production. And again, these are standard systems. We make the same systems over and over, which means we manufacture them more efficiently and deliver them significantly faster than conventional systems. So it's often half the time or less. They have large install bases and long track records of reliable operation. We've got a very active user group that shares best practices and regulatory strategies. Even though many of them are direct competitors, they've still found that there is much to gain by working together to advance the science. And then, as Joe was talking about in the previous presentation, sometimes the best strategy for scaling up is actually to scale out. You can start with a single work cell to meet your initial capacity, and then instead of building a very large production line that meets your peak capacity, you start small, and then as you need to, you add additional duplicate capacity. And that means that delivery is fast because it's a standard system, and then the qualification of the individual systems becomes much easier because you've experienced with the same process over and over again. You can actually compress your qualification time down significantly on repeat installations. And again, because they're identical, fill recipes and parameters can be shared between systems, even if they're at different sites. So, having a look at those standard systems, so what does this mean for people filling mRNA LNPs or other LNPs? And let's have a look. The answer might actually surprise you a bit. So we talked to industry leaders last year, and we found that the biggest concern of, when it came to aseptic filling was to manage risk. Obviously, controlling risk to the patient was the highest priority, but project risk was almost as significant as a concern. And setting up an aseptic filling operation was seen as a very risky project. And the reason it was perceived as risky is that manufacturing leaders thought they needed a custom solution to meet the specific requirements of their unique drug product. everyone who's developing a new drug product is usually developing something unique. There's something novel about it. But custom is risky. You are, by definition, trying something that hasn't been done before and has an uncertain outcome. So we had these industry leaders have a closer look at the fundamental aspects of the filling process. Because after all you really need to do is put liquid into a container. The trick is, of course, is you have to do it perfectly every time. And so once they realized that the requirements were so similar, they concluded that they could reduce risk by choosing a standardized solution. So the answer to the question is what you need to consider when you're filling mRNA LNPs is kind of not much. A standard solution will likely work for you. So now that I've said that, what am I going to talk about for the rest of my time? Well, let's talk about some of the considerations for the processes directly adjacent to aseptic filling, for formulation and post-filling activities that actually do have some specific considerations for mRNA and other LNP formulations. To have a look at them, particle size can have a considerable impact on manufacturability. Filter selection and configuration, there's some important considerations there as well. Holding studies for temperature and agitation and understanding that are going to be very important. Container selection, your primary container selection, the materials, the configuration, what you're using for closures, has a very big impact on product stability and shelf life. Visual inspection has some complications that need to be considered. And of course, if you're developing something during a pandemic or anticipating a pandemic response, there's other things that you need to consider. And just a note, that's a picture of Moderna's clinical COVID-19 vaccine that was manufactured on a Vanrx SA25 at their facility in Norwood, Massachusetts. So the first thing we're going to talk about is particle size. And you have to remember that lipid nanoparticles are particles. And as a general rule, smaller particles filter better than larger particles. So your nominal particle size is generally going to be around the midpoint of your size distribution. If your particle distribution is very broad, you may run into issues filtering LNPs above 120 nanometers. The more you can control your distribution and keep your mean particle size below that threshold, the more likely that you will be able to effectively sterile filter your drug product. Now, of course, you may have a drug product that requires larger particle size for other reasons, clinical efficacy, whatever those reasons are. In that case, you're going to need to develop an aseptic process from the encapsulation step. That means that the incoming raw materials for encapsulation will need to be sterile when they enter in that process, either sterile filtered or provided aseptically. And the product needs to be kept in aseptic conditions from that point forward. This is going to mean, or could mean, a greater reliance on pre-sterilized components, disposable flow paths, vessels, other components that are going to be used in the process between encapsulation and aseptic filling. And that's going to increase your cost and your process complexity. And if you're not able to perform all the steps in a closed system, that's something that goes from encapsulation to aseptic filling in one closed assembly or system, you're likely going to need to make extensive use of aseptic connectors to hook up one process step to the next, or tube welding, some way of making a sterile connection between the steps. And qualification of this connection process is going to be extremely important. It's much more important than it is in an upstream biomanufacturing environment, because here you're dealing with the final product that will reach the patient. So the qualification is going to be even more critical at this stage. And so it will require significant effort. And so, looking at that, particle size really can have a very large impact on manufacturability. So you should consider those aspects when you're doing your drug development, the design of your drug product candidate. The other one big consideration is LNPs have different requirements for filter media. Polyether sulfone, like in Pol Supor filters, has been used very successfully for a number of LNP drug products. And so they're a good starting point. But you may, in fact, need to consider other filter media depending on your specific drug product composition and characteristics. And so this qualification can require much more work than traditional aqueous formulations. So you've got to be prepared to spend some time on this. It's a considerable amount of work. And as I'll mention in one of the other points, you're probably going to want to do it with two different filters so that you have something to fall back on if one of those filters isn't available. But one thing I will recommend is the use of a single sterilizing filter versus the use of redundant filters. Redundant filters are often used in large-scale filling where the filters are used inline in the process and can't be truly integrity tested until the end of the batch. So if one filter fails that post-use integrity test, the idea is the second one provides assurance that the drug product was successfully filtered. However, this filter integrity failures have a very low chance of occurring. And so this approach is complicated and wasteful, both in the fact that you're using an extra filter each time and also that you're going to have likely a little more inline loss via filtration. And of course, drug products aren't made any more sterile by filtering them twice. Once is enough if it's an effective filtration technique. So there's some strategies that allow you to use a single filter that give you the equivalent level of sterility assurance. The smaller scale batches, and this is done by many of our customers, the entire batch can be filtered prior to filling. So allowing you to then, do the integrity test on the filter, confirm that it was integral during the process, and then proceed to the aseptic filling with a very high assurance that of course everything has been filtered correctly. This is also a good practice if you need to keep your bulk drug solution or formulation chilled for product stability during your filling process. Filtering a cold solution can be a difficult process, and it's usually much more efficient to filter at elevated or room temperatures. And then you would chill into your chilled vessel, which would of course need to be pre-sterilized. You may want to use a disposable vessel for that. And then chill it and then proceed with your filling. And you can do the filter integrity test, get the go-ahead, and then go ahead with filling. And so this brings up the importance of hold studies. Many LNP formulations or drug products do have some sensitivities to temperature as well as to agitation rate. So if you're going to be chilling your bulk and agitating it during the filling process, you want to know for exactly how long you can do that for and how stable your product is. Aseptic filling does need to be performed at or close to room temperature. It's not effective to try and chill down an entire filling line. It uses a lot of energy, and it also introduces a large number of complications to the process. It'll affect your decontamination processes and has the possibility of introducing condensation and other aspects into the system. So it's something that's generally not done. So you're going to be filling at room temperature. That means you have to know the residence time of the drug in the system, and in other words, how long an individual vial is going to be exposed to room temperature, and whether your drug product can tolerate those temperatures. The nice thing about an SA25 is because everything is kept in nests, the residence time, this also applies to a microcell, the residence time is very, very predictable, and you can then design your filling process with that in mind. The other aspect, of course, is in your holding studies, you also need to make sure that when they are returned or your product is returned to chill conditions, it hasn't affected the product characteristics. Because you may in fact need to return to chill conditions a number of times. Again, visual inspection and labeling are also likely to be done at room temperature. So you need to be able to set aside time for that and ensure that holding them or doing those operations at room temperature doesn't affect your overall product quality characteristics. And then, of course, container selection. Choosing the correct primary container is extremely important, especially for products that are stored frozen. So we have customers using glass vials for long-term storage of products at -80 degrees Celsius. But COC, with cyclic olefin copolymer, which is a type of plastic, seems to be the preferred material for low-temperature storage. It's much more durable than glass and less prone to breakage. Now, glass generally has a better barrier for gas transmissibility, but there are COC vials that are available with a layer that prevents gas transmissibility during storage. So again, they're a very good solution for low temperature storage. And you may be looking at a picture. The vial in the picture looks quite different from normal vials. It has a plastic press fit closure instead of a traditional aluminum crimp seal. Now, the reason it has this is because the press fit closure is has more reliable residual seal force than an aluminum crimp. So they're compatible with standard serum stoppers, and they're also available pre-sterilized in the kit. Hi, John. and so stopper selection should also reflect your storage conditions. So if you are storing it frozen, you may need to look at different materials, or you may want to look at a thicker stopper to have additional compression when it gets into a frozen state. If you're at -80 degrees Celsius, you're generally below the glass transition temperature. So you want to make sure that at those temperatures, you're going to be maintaining your seal. So a thicker stopper flange can be a solution to that, and that'll help you maintain your seal integrity at lower temperature. You will need to do CCI studies or container closure integrity studies. And you may want to do them while the vial is in a frozen state. And that's going to be difficult using traditional methods such as dye ingress or bacterial ingress. So we'd recommend headspace analysis, where you're looking at the composition of the headspace gas. You can fill or put in an inert gas or semi-inert gas like carbon dioxide or nitrogen or helium, and then analyze the composition to see if there's been any changes that's been exposed to the environment. And so this is a non-destructive test, and it can be performed very rapidly without basically having your product thaw. Typically, it can be done in a minute or so per container. So there's very little impact on your product. One other area that you should pay attention to is visual inspection. Many LNP drug products, and this is one of my favorite words, are opalescent. That is, they're somewhat shiny, as well as often being translucent. So when you're doing the visual inspection, which is critical to your release process, you need to inspect for damage to the container and the presence of foreign materials. So as far as damage to the container goes, of course, because on Vanrx systems, you're using nested vials or nested syringes or containers, there's much less chance of damage to the containers during the process. In fact, we have customers that have very, very low reject rates, greater than 99 point whatever acceptance rates, which is extremely high for the industry. But the physical properties of LNP formulations can make it more difficult to detect particles. So they can cause false positives when you're using an automated vision system. So you may need to adjust the settings or adjust the detection algorithms so that it's appropriate for the actual formulation. And if you're using human inspectors, you may require extra time or even more than one inspector to look at individual vials just to make sure that they catch any particles. Again, it can add some time to the process, so you need to accommodate that in your holding studies or your residence time studies to make sure that your product isn't affected by those extended periods of time. The other thing that I kind of alluded to earlier is if you're working on a pandemic response product, either for this pandemic or a future pandemic, and I'm hoping that's far in the future, there will likely be constraints on your pharmaceutical supply chains. We've seen this in the current situation. It's an industry-wide problem and is being most felt in single-use assemblies, the pre-sterilized assemblies that are used in all aspects of the process, in the manufactured components that are used to make the assemblies, in the raw materials used to make the components, and we're also seeing constraints in the sterilization process for those assemblies. As well, for LNP formulations, you're going to want to look at your custom lipid supplies, make sure you've secured those, and any other components that you may be insourcing. You want to make sure that you have multiple suppliers. And not only do you have multiple suppliers that you've sourced, but you've qualified them as well so that you're not going to be held up. And the most important one to consider, of course, is the sterilizing filters, because that's the critical piece of equipment that ensures that the patient is getting a safe drug product. So you're going to want to qualify multiple filters so that you won't be disrupted in your production during a pandemic. And that actually applies to people that are just producing pharmaceuticals in the middle of a pandemic. You have to go through these same rules. So plan ahead, source multiple materials and fully qualify them. And order well in advance and provide projections to your suppliers of your future needs so that they can plan. The more advance notice you provide people, the better they'll be able to respond to your needs. And yeah, just a reminder, a pandemic is not the time to have a just-in-time inventory strategy. It's much better to plan ahead and make sure that you have the resources to make your drug products when supply constraints occur. So as you can see, even if a standard solution is effective for aseptic filling of mRNA LNPs, there's still some very specific considerations for the adjacent processes. Very happy to take questions about either the Vanrx aseptic filling work cells or the considerations for mRNA and LNP filling.